of tens or thousands of scientists from the various disciplines in both from the public and private sectors worldwide. One of the greatest impacts of having the edge sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or a few genes at a time with the whole genome sequence and new high throughput technologies we can approach questions systematically and on a much broader scale. This can study all genes in the genome, for example, all the transcripts and the particular tissue organ or tumor, or how 10,000s of genes and proteins would work together interconnected networks to orchestrate the chemistry of life. As stated in preceding section, 99.9% of the human genome among the human is the same. It is different sequence of DNA which make every individual unique Indian phenotypic appearance. If one aims to find out the genetic difference between the individual or among individuals of a population, sequence of DNA every time would be daunting expensive. Imagine trying to compare two sets of 3 into 10 to the power 6 specific DNA. Fingerprinting is a very quick way to compare these sequences of any two individuals. DNA fingerprinting involves identifying difference in some specific regions in DNA sequence called a repetitive DNA. Because in this sequence, a small set of DNA is repeated many times, this repetitive DNA is separated from a bulk genomic DNA as different peaks during the density gradient centrifugation. The bulk DNA forms a major peak and other small peaks are referred to as satellite DNA. Depending on the base composition, 80 rich or GC rich length of segment or number of repetitive units called satellite is classified into many categories such as microsatellite, meaning that these sequences normally do not code for any product but they form a large portion of human genome. These sequences show higher degree of polymorphism and form the base of DNA fingerprinting. Since DNA from every tissue such as blood, hair, follicles, skin, bones, saliva, sperm from an individual, so the same degree of polymorphism they become very useful identification through forensic application. Furthermore, as the polymorphism and inheritance from parents to children, DNA fingerprinting is the basis of paternity testing in case of dispute in polymorphism and DNA sequence is the basis of genetic mapping of human genome as well as of DNA fingerprint. It is very essential that we understand what DNA polymorphism means in simple terms. Polymorphism varies some genetic labor arises due to mutation. New mutation may arise in individual either in somatic or in germ cell that generate gametes in sexual reproduction organism. If germ cell mutation does not seriously impair the individual's stability to have offspring who can transmit the mutation, it can spread to the other members of population through sexual reproduction. Alice again recall the definition of Alice. Sequence variation has traditionally been described as a DNA polymerase. If more than one variant allele at locus occurs in human population with a frequency greater than 0.01, in simple terms, if an inheritable mutation is observed in a population at a high frequency, referred to as DNA polymorphism, the probability of such variation to be observed in non-coding DNA sequence would be higher as a mutation in the each sequence may not have any immediate impact in an individual's reproductive ability. This mutation keep on accumulating generation after generation and form one basis of ability polymorphism. There is a variety of different types of polymorphism ranging from single nucleotide changing to very large scale changes. For evolution and speciation, such polymorphism play very important role, and you will study this in detail at a higher classes. The technique of genotyping was initially developed by Unlike Jeffrey, he used a satellite DNA probe that shows very high degree of polymorphism. It was called as variable number of random repeats (VNTR). The technique, as used earlier, involves southern blot hybridization using radio label VNTR as probe. It includes isolation of DNA, digestion of DNA by restriction endonuclear separation of DNA fragments by electrophoresis, transferring blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. Hybridization using label BNTR probe Detection of hybridized DNA fragments by autoradiography. The BN belongs to class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged randomly in many copy numbers. The copy numbers varies from chromosome to chromosome in individual. Number of copies show very high degree of polymorphism as the size of BN varies in size from 0.020 kb concentrated after hybridization BN probe. The autoradiograph gives many bands of different sizes. This bands gives a characteristic pattern for an individual DNA. It differs from individual to individual in population, except in case of monozygotic identical twins. This sensitivity of the technique has been increased by use of polymerase chain reaction. Consequently, DNA from single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, there's a much wider application such as determining population and genetic diversity. Currently, many different probes are used to generate DNA fingerprints.